Hello, my name is Alexis Polkowicz, and today I'm going to talk to you guys about Apoquel's influence on the canine allergy immune response. So, some relevant background information. I'm sure that a lot of you have encountered dogs or maybe even cats in your life that had suffered from allergies during their lifetime. These allergies can be seasonal, they can flare up during the fall or spring, just like in humans. They can be breed specific. I know pit bulls are the infamous allergy breed. And these symptoms can worsen with age. And we'll kind of talk about why and how that happens later in the presentation. And the good news is that within the last few years, a new medication was released that could actually help dogs that were suffering from these chronic allergy symptoms. And that medication is none other than Apoquel. So just to give you guys a little foreshadowing of the things we're going to talk about that related to class, the first thing is that we talked about cytokines a lot throughout the duration of this course, and just for a refresher, cytokines are a class of small proteins produced by cells of the mammalian immune system, used as messengers in controlling the strength and duration of the immune response. We also touched base about what causes allergies in Lecture 5, and that was when a harmless substance is deemed harmful and the body attacks it. And the last thing is we went over how the JAK-STAT pathway works in Lecture 6. And it was really cool because I was a little confused about how this pathway worked going through the lecture, but this presentation actually made it more clear to me because some of the articles that I read explained it a little bit more in depth and applied it to medicine, which was really interesting. Before we can talk about how Apoquel works in the body, we have to really understand what allergies are. And as stated earlier, these can arise when your body deems a substance that isn't harmful at all, such as pollen or dust, foreign, and your immune system actually creates antibodies to try to fight off this allergen. Responses to these allergens can be minor and may clear up with taking some medication like Zyrtec, or just simply going inside and distancing yourself from the pollen, or they can be severe where people may have to go to the hospital because they can't breathe. Repeated exposure could worsen symptoms, and as mentioned in the first slide, this is because the more times that you're exposed to these, quote, harmful substances, the more antibodies your body produces to fight them. So the more times that you're exposed to them, the more dramatic your immune system's response is going to be to try to fight them off. Okay, so now we can talk about the really interesting stuff, which is how Apoquel actually works in the body. And Apoquel works by selectively inhibiting certain JAK enzymes, which prevents signals being triggered by these JAK enzymes that are involved with itching and inflammation in these allergy patients. So by inhibiting these JAK enzymes, Apoquel actually decreases the activity of itch-inducing and inflammation-causing cytokines, such as JAK1 and JAK3 dependent cytokines. And these are the two JAKs that Apoquel targets. And what's interesting and unique about Apoquel is that it does not impact JAK2 dependent cytokines. And these are the guys that are involved in the production of new blood cells and even innate immune function. So we need these guys. So it's nice that Apoquel doesn't really interfere with them, and it just targets JAK1 and JAK3, which are the mean little buggers that cause the itching and inflammation to occur. And by inhibiting these JAKs, this prevents signals that promote infiltration of inflammatory cells into the dermis, which in turn prevents itch neurons within the skin from transmitting the itch signal to the brain. I would also like to add that in PowerPoint, my labels here are not messed up. I think that was just a glitch uploading it into Explain Everything. The first study that I'm going to talk about was actually carried out at a Zoetis laboratory, and the objective was to determine whether the JAK inhibitor could reduce the activity of cytokines implicated in canine AD, which is atopic dermatitis. The methods used in this study were basically researchers took active kinase domains for JAK1, JAK2, and JAK3 and used isolating enzyme assays via caliper microfluidics technology to determine how potent Apoquel actually was against each of these JAK family members. And the caliper microfluidics technology allowed these researchers to thoroughly analyze samples in seconds rather than minutes and helped to improve their efficiency. It also eliminated the need for these researchers to handle time-consuming SDS page gels. Shout out to my biochemistry colleagues, as we all know how painful these were. These tests were performed on 38 different non-JAK kinases as well, to see if this fancy word right here, which is just a fancy and scientific word for Apoquel, they were just testing to see if Apoquel had these same effects on kinases that were not part of the JAK family. All of these tests were run in duplicate just to ensure that their results, to make sure that no mistakes were made on their part along the way and that their results were accurate. This study actually didn't have any figures that stood out to me. This one was actually the one that I chose to present because I liked how it broke it down into a nice and easy to read chart. But their overall results found that Apoquel was a selective inhibitor of JAK1 dependent cytokines, and these guys are the ones that, as we stated earlier, are involved in allergy, inflammation, and itch sensation, which suggests that these are the mechanisms that Apoquel effectively controls these clinical signs through. And this was big because, yes, I told you guys that at the beginning of the presentation, but research had to be done to figure that out, and this was one of the founding articles that established that Apoquel works by working on the JAK1 dependent cytokines, and I thought that was really interesting because this was groundbreaking. The second study I'm going to talk to you guys about is one that was done in practice to try to see how safe this medication is on dogs. And the objective was to evaluate the efficiency and safety of Apoquel for the control of canine atopic dermatitis in a randomized, double-blind, placebo-controlled trial. And the methods of this study, which I liked, was they actually involved the pet owners, so this made them feel like they were active participants in science, which they were, which was cool. So dogs were randomized to receive either Apoquel twice daily for 14 days, then once daily for up to 112 days, or a placebo medication for the same thing. So they would get the placebo medication for 2 times a day for 14 days, then once a day for up to 112. The owners were able to assess how much their dogs were itching in what's called a visual analog scale. So they just logged how much their dog was itching on day 1 compared to day 84. And throughout, they can see if the itching subsided or if it stayed the same, so on and so forth. Clinicians also scored the itching that they perceived on the dogs during recheck examinations via a canine AD extent and severity index. After day 28, all placebo-treated dogs had moved to an open-label study, which basically just meant that they were told, the owners were told, hey, you're giving your dog the placebo medication, so that's why your dog is still itching. So that kind of made the results after day 28 be a little bit biased because owners knew what they were giving their dogs, so they were more likely to lie about what they were seeing. And if you took any neurology classes or anything like that, you would learn that this is a flaw in a lot of studies that do this. So as for the results for this study, the figure on the left actually corresponds to this second bullet point. This is the doctor's ranking of what they were observing, what was happening. So as you can see on days 14 and 28, there was a 48% reduction, meaning that the dogs quit itching so much, compared with the 1.7% reduction and a 3.6% increase in the placebo-treated dogs. So this meant that the placebo dogs didn't really quit itching, and some of them even started to itch more. The differences were very significant at all time points, and the overall results showed that Apoquel was actually able to provide rapid, effective, and safe control of these itching symptoms, because none of these dogs exhibited any concerning clinical side effects during this trial, which was very promising. The first figure in this study talked about, or was basically the same exact figure, but it obviously was the VAS diagram that showed the owner's assessments, which in my opinion they did a pretty good job because it looked almost exactly the same as the doctor's visualization scale shown here. The third article that I'm going to talk to you guys about is actually almost the exact same kind of study as the one we just talked about, but it is in cats. And I thought this was really interesting because cats don't have any allergy medication out there like dogs do. And I know that this whole presentation was supposed to be about Apoquel's effects on dogs, but I included this article because I found it really interesting that one day Apoquel may be approved to be used on cats. And it's also promising because the study was extremely recent. It was done in May of 2019, which means that it may not be long before I myself am prescribing Apoquel to a cat. So the objective was to evaluate the safety and clinical effects of Apoquel in healthy cats. The methods were carried out in a laboratory, so these were lab cats. There were 30 mixed breed cats, and their weights are listed. Each were randomly selected to 3 treatment groups of 10 cats each. Cats in 2 of these groups received Apoquel at a smaller dose, and the cat in the other group received Apoquel at a slightly larger dose. And cats in the third group were given placebo tablets. This study, unfortunately, was yet another one that didn't really have any figures in it, so I chose to show Table 1, which was the most relevant to what the results of the study was, which is that Apoquel was very well tolerated by cats that received both the smaller and the higher doses of Apoquel, which means that this medication appeared to be safe for this species when administered twice daily for 28 days. The study made sure to say that more studies will need to be done before this medication can be promised to cat owners that are suffering from chronic allergies, just to ensure the safety and especially for long-term use in cats, because nothing's been done testing that out yet. And as you can see from the table, no dangerous symptoms were observed in cats that received the smaller dose of Apoquel, and only a few of the cats that received the higher dose experienced things such as vomiting or soft stool. I should here. But overall, the feline blood cell levels remained good, their kidney liver enzymes remained healthy, and the urine-specific gravity and pH in all of these felines remained normal. The only thing was that there was a significant increase in fructosamine levels observed in both of the treated groups as compared with the placebo group. However, these fructosamine levels remained within the normal reference range. They just increased enough that the researchers had to make note of it in this study. Here's a quick summary about all 3 of the studies that I mentioned, and their key findings are listed. All of these studies emphasize how effective Apoquel is at calming these allergy symptoms, itching and inflammation in dogs and even cats. There's a small plot twist. Some future directions would be to perform more studies in cats so that they too can have a promising, effective, and safe medication for allergies and itching. And now that I'm thinking about it, there weren't a lot of studies that mentioned long-term use in dogs and cats. So I think more studies that focus on the long-term usage and the potential side effects that can develop from being on this medication over time would be interesting to look into and see if anything would happen in the long run, or if this truly is a genuinely safe medication for these animals to be on long-term if need be. Here are my references, and otherwise, happy summer, everyone!